Hello, and welcome to Rooted in Research, where evidence, expertise, and real world perspectives come together in one space. Hello, my name is Sang Choi, pharmacist and research scientist at the Office of Cannabis Management. And we are so glad to have you with us today. And I'm really happy to be your moderator for today's webinar. So, joining us today is Dr. Harold Hahn. He is a scientist, an entrepreneur, and the founder of Vertosa and PhytoRx, which are companies leading innovation in how plant-derived active ingredients are delivered, dosed, and experienced. He holds a PhD in surface chemistry from New York University and is the inventor on multiple patents in emulsion and delivery system technologies. His work in colloids and nanoemulsions has helped bridge complex chemistry with practical solutions, turning theory into stable, scalable, and predictable formulations across fields like medical diagnostics, food systems, and plant-based therapeutics. Prior to founding Vertosa and PhytoRx, Dr. Hahn served as a senior scientist at Biorad Laboratories, developing emulsification oils used in the digital droplet PCR platform, which is a technology that now plays a vital role in early cancer detection and personalized medicine. And in the industry, he is affectionately known as the happy chemist, and I think you will see why. Dr. Hahn blends scientific rigor with practical insight, bringing consistency, efficacy, and trust to plant based products for clinicians and consumers alike. But before we get started with today's programming, I would like to take a moment to share a brief disclaimer. The Office of Cannabis Management makes no claims, promises, or guarantees about the accuracy, completeness, or adequacy of the content of this video live stream event and expressly disclaims any all liability of the content and for errors or omissions in the content of this video live stream event. This video live stream event is for informational purposes and convenience of the public only and does not constitute endorsement, recommendation, professional advice, or favoring by the Office of Cannabis Management. The content of this video live stream event is based on information available at the time of recording and is subject to change. And with that, we are very excited to hear Dr. Hahn in his presentation on cannabis emulsions, transforming consumption, therapeutics, and research.
All right. Thank you, Sam, for the introduction. And everybody, this is Harold. I'm so honored to be here today. I know we may have some technical difficulties about some internet lagging. Can everybody hear me okay? Is it okay for me to go? Cool. I'll take it as a yes. As a matter of fact, I'm a chemist. I'm an emulsion chemist, studying how to put oil into water and stabilizing them. And I've been doing that for 20 years. And really lucky to find application. I want to share some of the learnings we had of cannabis emulsion, how it transforms cannabis consumption, therapeutics, and research. Okay, let's take a look of the evolution of medical cannabis. Before 2000s, when patients only have the flower, right? The flower still worked, but it changes from batch to batch, is variable. And the issue with the flower is that patients may have to smoke many joints to get to the milligrams they need. And also, this picture of a patient smoking a joint just doesn't make sense. Rick Simpson, he invented the RSO oil. And this oil solved the concentration issue. If a patient now need a half a gram or a gram of cannabinoids, they can get it. But now this oil has limitations, such as it's pretty nasty if you consume it daily, and it's hard to meter dose. And in 2016, 2018, FDA approved drugs based on cannabinoids, such as dronabinol, Epidiolex, and this solved the meter dose issue. right? But then FDA only allow drugs that is single molecule. It does not. But if you look at today, the challenges in medical cannabis industry, I list a couple of them. I want to see if any of those resonate with you. Dispensary is not pharmacy. We love our bartender, but they're not trained as pharmacists. So there's a lacking of service to the patients. There's still too much focus on THC in a dispensary, not enough focus on CBD, CBG, other cannabinoids. And most consumption vehicles have their compromises, such as smoke, sugar, and then product has variabilities. For example, the flower may change from batch to batch, but also if you're living in California or Massachusetts, the state-run program, the product, the regulation are all different. The products are usually difficult to titrate, so it's quite difficult to be inconsistent dosing yourself. And then if you're taking a high-potency tincture or a fecal oil, it always comes with a bad sensory, and usually sometimes with a poor PK profile. And the product right now is lacking the customization. What you get is what you get. I see people already, smoking a joint while getting some tinctures and some gummies. That is the way they can customize for themselves. And then if we go through FDA pathway, it's usually not compatible with full spectrum. It's single compound. So before I propose a potential solution, I want to imagine, what if we can drink cannabis? What kind of features can this bring benefits to the patients? So in the following slides, I want to show you some of the studies and thoughts we have. So a transformation is needed because we all know every cannabinoids, they are oil soluble. They don't go to water by themselves. So there is going to be a transition that is needed. And that transition is, different technologies can do this job to introduce cannabinoids into water. And this table summarizes some of the main technology. For example, you can leverage a liquid emulsion, such as nano emulsion or micro emulsion, order from spray drying, spray that emulsion onto a powder substrate and then go to the water, or co-crystallization, which is you put your cannabinoids onto a substrate directly, which is water soluble. And then I also seen people doing co-solvent using ethanol as a solvent to introduce cannabinoids into the water. And those methods are keeping the cannabinoid structure intact, but also you can change the cannabinoid structure, such as you can put a sugar molecule onto CBD. So you can make the CBD water soluble, which is called glycosylation. But this technology will alter the CBD structure, making it a prodrug protocol. So right now in the market, nano emulsion is infusing over 90%. So it is the most used technology. And people often ask me, what is the difference between a nano emulsion versus a micro? Oh, it's about the size, right? Nano emulsion must be smaller droplets than micro emulsion. However, it's less about the droplet size, but it's more about the energy input. So we draw this picture to show you the true difference between nano emulsion and micro emulsion. So we'll start from the middle. The middle shows you water, oil, and emulsifier. So to make a micro emulsion, you add a little bit energy into the system, this little bump, and then all the composition will find its own way to be stabilized. So it goes to a lower energy state to be stabilized. And so it's called self-assemble, right? You don't need to put a lot of energy in this. So it's quite easy to make. However, the compromise with this is usually you have to use many excessive amount of synthetic emulsifiers, right? They are very effective in making macroemulsions. Well, if we start with the same original point, but if you add energy into the system, which is the bar here, now you're making a nanoemulsion. The nanoemulsion, you have to use a certain equipment, but the benefit is you make it more versatile. You can use different emulsifiers. You can make small droplet size versus you can make a big droplet size, right? You can design the emulsion to a certain application. So that's why size is not the main difference between the two emulsion, is the energy input during the process. So how do we make a nanoemulsion? You always start with the chemistry, which includes cannabinoids, carrier oils, emulsifiers. And then you have to add energy into this. We call physics, right? Called processing, energy input. Those energy input will split the droplets and produce the droplets. And then in the end, we have to do quality control, including droplet size distribution, potency, microbial, heavy metals, solvent, pesticide, microtoxin, pH, density, rheology, and sensory. And that's at our company, every batch go through this protocol before we go out the door. So what does a nanoemulsion look like? If you open our fridge, you may see, jugs like this. And different colors means different emulsion formulas. And each of the food jug can potentially infuse 500,000 cans of 5 milligram per can. You can go into the jug and you will see trillions of those small droplet size floating among them. And they contain either CBD or THC or any other cannabinoids. And in order to be stable, we allow them not touching each other, not merging with each other. They are intact with themselves. So this is what a nanoemulsion looks like. So how do we define a nanoemulsion? What kind of features it will give to our clients and the consumers or patients? So we put 5 pillars: stability, manufacturability, certification, and experience. Let me explain. Flavor is first of all, it's the food ingredients. It needs to be safe. It needs to be tasting good, right? So we have developed SOPs to test how to evaluate flavor because it's subjective. Stability means the emulsion needs to be stable, and then the infused product, such as gummies or beverages, they need to be stable to produce those emulsions, right? How scalable we can produce them. And then other perceived values such as label claim: is this synthetic, organic, natural derived? What is it looking cloudy? Is it looking transparent? Does it have a USDA organic certification? Is that GMP certification? And then the last piece is experience. This is the holy grail for pharmaceutical companies when you talk about PK and PD, because these ingredients will be absorbed by our body and give an effect, right? So we need to understand how effective it is in our body, what kind of effect the consumer and patients can feel. So of course, you want to design an emulsion that maximizing on all 5 pillars, but it's not really the reality. In reality, we need to understand how to design the emulsion to target a pillar that is mostly desired by our clients and by the consumers. So give you an example. If you use a synthetic emulsifier, right, the droplet size may be smaller. So you decrease a little bit here because you use synthetic emulsifiers. The small droplet size will change the flavor, will change the onset, which is the experience, may impact the stability and also manufacturability. So the key is we need to understand not only how to measure each pillar, but what kind of lever we can control to adjust each pillar. So I want to talk about the aging between THC emulsion and alcohol. because we know liquor, alcohol, you can store this in the basement for 2 decades without losing the alcohol content, because ethanol is an inert molecule. But THC is very finicky. It always look for ways to get lost, either physically or chemically. So, for example, chemically THC oxidizes into CBN. So we really need to pay attention to the stability of emulsified cannabinoids. So in order to understand how quickly the potency can get lost, especially in the water form emulsion, we did the study. We put 20 milligrams of emulsified cannabinoids into 12 ounces of water at ambient oxygen, so high oxygen, and we put it at 40 degrees C for 1 week and 2 weeks to measure the potency loss. And you can clearly see that all 6 cannabinoids belong to 3 categories. THC and THCV, they are the worst. They can lose potency really quickly. Well, CBD and CBG follow them. So this table tells me 2 things. One, if you are making a THC-infused beverages, then you should be really careful about maintaining the oxygen level. And 2, if you are making a THC beverages versus a CBD, the way they oxidize. Cannabis emulsion can also bind with natural sediments. For example, this is a kombucha base, and you have sediments here. And when we add emulsion into the base, after sediments form here, we take a sample from the bottom, we take a sample from the top, we perform potency test, and we see that the ratio from the bottom to the top really changes. If we're using water, there's no change, it's 1, right? So the top and the bottom has the same potency. But if you do it in a kombucha, you see the bottom has much higher potency than the top. It means that the sediments actually physically binds with the emulsion droplets and drag them to the bottom. So we need to pay attention if we produce a heavy sediment containing beverages, right? And there are protocols we already designed to produce this, but this is an effect that can impact the potency. Can liners. This is a topic that is has been haunting our industry because in the aluminum can, there is a liner like that. The liner is there to protect the liquid, to erode the aluminum, but those liners, they're hydrophobic, meaning they like oil, right? So THC is hydrophobic. So there is a phenomenon called scalping, which means the THC molecule not only staying in the droplets, but also stuck to the liner. So if this happens, you have less potency in the liquid, but more potency on the liner, so consumer gets less active. So it's losing the potency. And why this happens, we have a theory that the THC molecule, we think they are staying in the droplets. Actually, no, they are constantly coming in and out of the droplets. This phenomenon is called partitional equilibrium, and it's very normal in all emulsion formulas. And if this happens, and if the droplets are close to the liner, there is a chance that the THC will get stuck onto the liner. So the scalping rates between the emulsion and the can liners have been measured for many years. And our company is one of the pioneers working with packaging companies to really understand what is the good combination between the chemistry of the emulsion and the chemistry of the liner. And this is the summarized slides, shows that there are 3 main liner types: acrylic, BPA, and BPA-NI, non-intended. And our emulsion shows high stability with BPA and BPA non-intended, but low stability at acrylic liner. So knowing this is great, right? Knowing this will help our clients to avoid the liners they don't want to use, right? But those liners really look similar by eye. right? It's visually they look similar. So how to tell the liner apart to help the industry? Well, we developed, with the help of Enresco Lab, we developed a method. It's called FTIR. And essentially, you cut the liner, you put the liner under IR, you detect the spectrum of the lone liners over here. And then for any parameter, is this liner compared to other known liners? And the software can do you a favor and give you a like percentage similarity. For example, this liner is 99.1% similar to V70Q11 liner, right? By running this test, now we can build more confidence into the liner we use, so that every step, every data can add confidence to the consistency of the beverage potency. Okay, so our emulsion, like I said, is a food ingredient first, but it also follow a pharmaceutical-like standards, which is the droplet size distribution. So this is the droplet size on the x-axis, and every batch of what we deliver from our company is following distribution like A, because if you have bigger droplet size, smaller drop, the flavor, the effect is not consistent, right? So the way we promise our clients and the patients is we want to make sure they get the same ingredients every single time, so they have the same effect. And then talk about the small droplet size. How does it impact the way we absorb THC in our body? This is what we did as a human PK study. This is me volunteering for the study. And we have 5 other people, so 6 people total. On 2 different days, we tested human blood testing of 15 milligram THC, one in the distillate-infused gummy, one in the emulsion-infused gummy. And we take the blood for the whole day, right? And then we average the data, and you can see that the emulsion-infused gummy quickly peaked before 10 minutes for all 6 people on average. And we have a higher Cmax. We have a much shorter Tmax to Cmax. So this is the average data, but I want to show you the raw data, which is interesting, that the 6 people, when they consume distillate-infused gummy, their Tmax ranging from 45 minutes to 180 minutes. But when they're consuming emulsion-infused gummy, their Tmax peaked before 10 minutes for all 6 people. So this shows that and then if we do an ANOVA analysis, we can see this Cmax, the Tmax is really significantly different between the 2 formulas. This is a big advantage for patients, because this means using an emulsion for patients can get a quicker onset for a quicker relief of their symptoms. So it's not only Vertosa doing the research. We do need to build a village of researchers to fully understand the emulsion PK and PD, and give a shout out to Johns Hopkins research, Tori Spindle and Ryan Vandery. And we are lucky to precipitate into their study. One of their study is to understand drink versus a brownie versus a gummy, emulsified versus distillate version. What are the uptake difference between the formulas? And you can see by 2 different potency, emulsified drink give a quicker onset, right, in both situations. And I know more researchers are getting into the study, especially after Schedule 3, potentially. And we know UCSF just won a grant to study the PK and PD profile of infused beverages. So I really hope more researchers are spending their efforts in understanding this, because millions of consumers will be benefit of knowing those insights. Okay, so we talk a lot about emulsion applying in the consumption route. When we look at still the main way, and we asked ourselves, is there a better way for patients to get their cannabinoids consistently and effectively? And this is a new concept that we are launching. It's called a PhytoRx. This is a glass bottle, Cannabis Emulsion, we fill it to a certain ratio, and there are 60 pumps into this bottle. Each pump will give you 50 milligrams CBD, 25 milligrams CBG, and 5 other minor cannabinoids. So, you can pump it into your tea, soda, juice, coffee, anything you drink at home. So it's a much easier way for patients to get their cannabinoids daily into a routine or a ritual. And people ask us, why you pick 50 CBD and 25 CBG per pump? What kind of condition you're targeting? Who can be using this? And in order to answer that, we create this 3-lane structure. We know our health is complex, our body is complex. So we hope this can explain this in a good way. The first lane is the actives, meaning they're all drug molecules, botanical or synthetic. The second lane, they are medical conditions, all the illness we have. The third lane is us, the genetics, right? We have 7 billion people, right? No two people share the same genetics. Okay, so the FDA synthetic pathway is one molecule for one condition for millions of people. And the benefit is very simple input and output on the 2 lanes, right? But then when you're expanding into many genetics, some people may be reacting to this compound really well. Some may not reacting to really well. Some may have severe side effects. So this is the inherent issue with this philosophy. And then over time, FDA also allowed botanical pathway, right? You can extract different compounds from the plants and then make into a medicine. So the benefit is instead of one compound, now you can have multiple compounds, but it's still targeting one condition for millions of people. And then we ask ourselves, many disease actually end with the same itis. What does that mean? Well, it means the inflammation is likely the root cause. And then we did our research. We see CBD and CBG, they have already proved, either, and we asked ourselves, why can't we leveraging the plant-based therapy and combining CBD and CBG, instead of treating one single condition, we are treating the root cause of those conditions, which is lower the inflammation. So this is the idea behind the PhytoRx application. So talk about emotion, talk about emulsifiers, surfactants. Some health-conscious consumers will be really worried because we know consuming, petroleum-based synthetic emulsifiers could have a bad effect on our health. So I want to introduce you what kind of emulsifier tree here is coming from. The Kila is coming from the tree here. It's called the soak bark tree down in Chile. The molecule is like this. It's a traditional emulsifier and is naturally derived, is certified organic, and has GRAS status. But not only that, when we dig into Kilaya, it's a very amazing molecule because paper has shown that Kilaya has immune stimulation property, anti-inflammation, anti-cancer, antiviral, antibacterial, and actually at certain concentrations, lower concentrations, it can promote the gut health. So selecting not only consumers but patients effect of the emulsifier. So in the end, if we zoom out a little bit, we are really combining different natural extracts from 3 plants: from the hemp plants, from the coconut plants, from the soak bark tree. The hemp plants give us CBD and CBG. The coconut plants give us organic coconut oil as a carrier oil. and the soap bark tree gave us kila extracts. And then we took those things and we put them together by emulsification, and then we diluted to a spec into a product. That is why the Phyto X is really a true plant medicine. Okay, so going back to the first question I asked in the beginning, how can drinking cannabinoids benefit patients? I think to summarize, it has a fast onset, proven by our human PK study. We have a high bioavailability, thanks to the small droplet size. Accurate dosing, thanks to the peak distribution, the tight distribution, and also the mechanism of the pump. Ease of use. We don't have to be smoking a joint or holding tinctures in the mouth. You can just drink it. Stable shelf life. Data to show that the bottle has 12-month shelf life when it's closed and 2-month shelf life when it's opened. No compromises, no smokes, no sugars. And then under the full plant, we have other cannabinoids in there. And highly customizable. This is the feature that is super important to me, and I want to expand a little bit on how Emulsion can help customize ratios. So imagine a future of personalized therapeutics, where our company, we already make to make the Phyto RX bottle, we actually combine 2 emulsions together, right? CBD emulsion and a CBG emulsion to target inflammation. But then actually we can leverage. So compared to a pill, which is really hard to customize liquid ratio to a certain combination is fairly easy, right? So this aspect could really open up a personalized medicine, and then just even from a single person over time, I may need to change my cannabinoid ratio emulsion here. Okay, the last 2 slides, I want to show you where we have seen non-emulsion can help cannabis research. For example, you can support the animal study. Typically, you have to do the garage, right? This method is really it's really inhumane. It stresses the animal out, and the PK can be variable based on the oil, and the outcome could be messy, right? But then what if we can use our non-emulsion and we give this to a standard kit to the researchers? They can just use pipette to whatever ratio they want to use, and then they can give this and let mice drink it. There's no stress, and they can drink it with a higher PK. So it's easy, it's clean, effective, repeatable, customizable, and we can do the PK. They can cite our PK profile, right? So we are working with certain professors in the country to see research. So this is animal research. But for human research, this is more interesting, because if we want to give cannabinoids to a clinical research to human, if we use tinctures, that's the only way now. Tinctures, the control may taste not bitter, but the active may taste bitter. And tinctures, people don't really accustomed to taking tinctures every day. And the tinctures is really hard to meter dose. And so recently, we've been working with some professors, and they want to give patients cannabinoids with control sample and active sample. And they say, Harold, can you emulsify this? I said, sure, we can. But then can you put emulsion into a glass bottle with a pump so I can tell you how many milligrams I want my patients to take every day, so that you can meter dose it, and the patients can just drink it into their coffee or smoothies. So that is an easy way for them to follow the study. I said, sure, deliver sample to the patients, and the beauty is you can make a blank sample. The blank sample is the control, which looks similar and taste similar to the active emulsion. So by doing this, it's very effective control sample. So for anyone sitting here who are thinking about doing clinical research using cannabinoids into a pump and deliver to your patients. Okay, that is all my slides. And the last one, I want to say thank you for OCN to offer me this opportunity, and thank everyone here to come in at 40 me open my mind that make me a better person, and I really appreciate my journey in the cannabis industry. I learned a lot, so thank you. Any
Dr. Han, thank you so much. Informative and all the innovation that's going on there. So I think at this point, we'd like to take some questions from the audience. So I think we'll start seeing them pop up here. All right, so let's take the first question. So what constitutes as a nano emulsion versus a non-nano emulsion? How do different emulsion formulations impact the testing?
That's a great question. In my slides in there, I compared non-emulsion versus a micro-emulsion. They are different, right? So people often think about the size because you imply the size, the word nano versus micro, but it's really the energy, like what I showed in the slides. So the detecting method really shouldn't change when you're detecting the raw emulsion, but when you put the emulsion into a product, such as a beverage or a gummy, certain beverage contain fat, lipids, and cream, those ingredients may impact the way you extract the cannabinoids.
Okay, thank you.
Hopefully I answered this question. Thank you.
We'll take the next question. Okay, so what is the expected onset time for the consumer? I know you kind of touched upon this, but maybe you can elaborate a little more. And is it faster than standard edible products?
That's a great question. So when we emulsify THC, we do a couple of things to it. One, we make it water compatible, so it can go to water. At the same time, we create so much surface area, right, compared to a big oil droplets. So that's why when you are drinking this, you absorb the THC mainly in the small intestines by mixed micelle, by formation of mixed cell. So if you fill your body with already emulsified THC, the path from that to mixed micelle is very short. That's why you have a very quick onset. So the onset to typical beverage consumer, we say it's mere beer. Before you drink this can, you should start to feel it, right? Doesn't mean you are really high, but you should start to feel there's a change in the mindset, right? So you can pace yourself. And this is the advantage of beverages. Two advantage. One, quick onset, pace yourself. And two is you can meet your dose. If you start to feel, okay, I'm good enough, I don't need to chuck this can along, I can just put it down, right? In terms of the gummy, if you eat it, it's in you already.
Right, you can't take it back. Okay, next question. All right, so when transforming oil-soluble cannabinoids into stable, water-compatible formats, would an expiration date be established, and would it have a longer shelf life?
That's a great question. Shelf life, depending on many different things. You can say microbial shelf life, potency shelf life, droplet size aggregation shelf life. And if any of those fail, that's your determination. So in terms of when we evaluate our shelf life, we do a bunch of testing. And one of the key thing is potency decay, because we know THC will lose potency by itself. Even a distillate, you will slowly lose potency. But if you emulsify into trillions of smaller droplets, you are opening up so many surface area, right? So that's why the storage condition the container you use in emulsion, and there's a shelf life to it. So the emulsion after it's being made, is the clock is kicking. So there is going to be a decay. But if you do things right, the decay, you can manage the decay. So currently at our company, we set 12 months shelf life from the day of production.
Okay, great. Thank you. Do emulsified products need to be shaken before use? Is there any separation over time? So this person is thinking about carbonated beverages and how this might impact potency or dosing.
Great. This is a great question. So, no, our promise to our clients is we want to give a homogeneous product to the consumer or patients consistently, right? So if you have infused beverages or the Fido X bottle, our promise is on the top, from the top to the bottom, the potency should be the same, right? Well, other things may change. For example, if you have a kombucha, heavy sediments, the bottom may have a higher potency than the top. That's based on the base difference. But our promise is physically, we should remain the stability of the potency. We don't want a consumer to drink half of the can and only get 20%, and the other half of the can have 80% of the potency. That is not good to build trust, right? So yes, our work, the way we're doing is we want to guarantee the potency homogeneity in infused products.
Yeah, very important. Okay. Is there any cost-effective way to make nano emulsions with the syrup?
Cost-effective way. To make nano-emulsion at scale, you need professional equipment, right? But if you're thinking about making nano-emulsion at home or a kitchen, there are, depending on how rigorous you want the quality to be. For your personal consumption or a small group, you can buy high shear mixers. You can buy some food grade emulsifier from online. I have seen my friend whipping up their formulas to make a bakery or make their own infusions. But to make a high quality standard non-emulsion, you do need standard equipments.
Okay. Does concurrent alcohol intake affect the bioavailability or absorption of the emulsions? Are there any foods that can affect the release mechanism?
Oh, this is a great question. Let's talk first, because there are studies already showing that when you are, this is a great question, because when you are drinking alcohol while eating a burger, you usually get less drunk. When you are drinking a cannabis beverage while eating a burger, you usually get higher effect. So there is a mechanism in the small intestines. When you're eating this food, your body is releasing biosalt, right? And the biosalt helping formation of the mixed mast cell. So that will help absorb the THC better, quicker. However, depending on the food, if you're eating a salad, which does not have a lot of fat, so the help of the absorption may be less, right? So depending on how much food you're eating, what type of food you're eating, there is or may not be contributing to the higher PK. So all I'm trying to say is, those are theories, right? Those are earlier science, but we do need scientists, researchers to study this. Like I'm really excited about UCSF winning this grant. They want to fully study the absorption of drinking beverages, right? So they can study different food, different amount of food. If you sip on it versus chug on it, if you drink 10, like 10 milligram versus 10 milligrams in the afternoon, is that double the concentration in the blood? So many of those are millions of American consumers would love to know, right?
Yeah.
So I'm excited for more researchers to come in and give us insights.
Yeah, that sounds great. I think you're right. I think a lot of people do want to know. Okay, we have two more questions. So how does emulsification work to enable a better delivery delivery in water-based products such as gummies?
Oh, this is a great question. Traditionally, gummy has a slow onset, right? If you just use distillate, some other brands and companies use phospholipid licensing to help gummy absorb a little quicker. But in our study, we have tried our emulsion, Kilaya emulsion. If you put that into the gummy, our PK data showed the onset is super fast, like within 10 minutes. And we have a client in Texas. They were selling distillate gummy to the PTSD veteran. And now they switched our emulsion gummy. And the most important thing for the veteran is for PTSD, they have this phase of coming up, this emotion, right? And then when they feeling these things coming up, they pop a emotion gummy, and quickly within 10 minutes, they can feel, okay, it's coming down. So I do see a lot of potential of using emulsion gummy to make quick onset as a industrial standard for gummy. But also a lot of research needs to be done. Kilaya is one thing we have, but our RD team is looking at different ways, different emulsifier combinations to put into the gummy. So more to update, but we do have one solution, but more, I want to research this more.
Yeah, great. Thank you. And last question. Please discuss the relationship of THC absorption in the skin.
In the skin, yeah, we have done some skin penetration study using CBD. So I don't know much about THC and CBD. It's different, especially in pension. But study, we find out if CBD is delivered by the emulsion versus CBD in a MCT oil, the emulsion will deliver CBD more deeper into the epidermis layer compared to the MCT oil. So the epidermis layer and dermis layer are different because one is defined as cosmetic, one may be defined as a drug. Because if you deliver THC or CBD into the dermis layer, which means you are going into the circulation in the blood, right? People may feel this. So how the nutrition is, they impact the regulations and the claims we make. So there are a lot of study, I'm not a skin expert, but I know, there are a lot of research needs to be done here to give us better answers.
Yeah. Well, thank you so much for answering all our questions. So as we conclude, we'd like to remind you that replay of today's session will be available on our YouTube channel immediately following the presentation. You can find links to Dr. Han's work and other resources in the chat. Please visit us at cannabis.ny.gov/research for more information about the cannabis research license. And when you have a moment, please take some time to fill out the post-webinar survey so that we can have more engaging conversations like this. And if you or anyone you know are interested in being one of our featured guests for the series, please complete the link to the form in the chat. And we will be offering the Medical Cannabis Program Practitioner course virtually on February 19th from 5 to 7 p.m. It is a 2-hour course that will provide overview of the endocannabinoid system, medical cannabis pharmacology, New York State medical program requirements, and more. It is intended for physicians, physician assistants, pharmacists, and other licensed clinicians who are interested in becoming certified and integrating medical cannabis into clinical practice, or if you're simply interested and you'd like to learn more about the medical cannabis program and medical cannabis and its clinical use, please join us. So we hope to see you there. And so please stay tuned for more information on registering for the course. Please stay tuned for our next Rooted in Research. And we want to thank you for joining today. It's really been a privilege having Dr. Han share his expertise with us. And, please stay curious, stay connected, and we will see you next time.